Let's first talk about some of the people that were important to figuring out that biological organisms are made of cells. First, we have Anton van Leeuwenhoek. He was one of the first to actually find single cellular organisms living in pond water. The picture that's here is his microscope. He took this contraption and there's a little pin here right behind an eyepiece that he placed a drop of pond water and looked at it and saw little things swimming in it. He came to realize that organisms were not just the plants and animals that he saw around, but he also came to see that there were small living things within a droplet of water. Then we have Robert Hooke. He looked at the bark from a cork tree, and he noticed that there were these boxes kind of all lined up, and it reminded him of a monastery, and the rooms in a monastery were called cells, kind of like a jail cell. He named these small little boxes cells, and that's how we got the name of cell. Then we have three different people. We have Schwann. He did a lot of research in regards to animals and looking at animal tissue and realized that animals were made of cells. Then we have Schleiden. He did very similar work to Schwann, but he did it with plants and realized that all plants are made out of cells. Then we have this other guy, Virchow. He noticed that new cells came from old cells, so old cells would divide to create new cells. This leads to what we know as the cell theory. The cell theory has three components. All living things are made of cells. We have animals and plants both being made out of cells. Now we know there's more than just animals and plants, but that all living things are made of cells. That cells are the basic unit of life. The smallest life can be is a cell. And that new cells are made from pre-existing cells. So new cells come from cells that are already here. We have two major types of cells. We have prokaryotes and eukaryotes. Prokaryotes are simple cells. They do not have a nucleus. The things they do have are DNA, and it is free-floating with the inside of the cell, a cell membrane, cytoplasm, and ribosomes. They may also have movement pieces such as flagella or cilia. They also might have a cell wall or a capsule on the outside, but they are fairly simple cells containing at least cytoplasm, ribosomes, DNA, and a cell membrane. Then we have more advanced cells called eukaryotes. Eukaryotes are the cells that have all the different organelles. One big difference between a eukaryote and a prokaryote is that eukaryotes have a nucleus. They have a wrapper or they have an envelope around that DNA. They also have all the different organelles in the mitochondria, the endoplasmic reticulum. They have all these different machinery parts in order to do their job as a cell. First, let's take a look at an animal cell. Animal cells are eukaryotic, so from here on out we're going to be talking about eukaryotic cells. We know that all cells have a cell membrane. The cell membrane is the outer wrapper of the cell. It is made of phospholipids, so it's made of a lipid that has a phosphate attached to it. We have cytoplasm. Cytoplasm is the area basically between the nucleus and the cell membrane. It's the liquid inside the cell as well as all of the organelles. We have ribosomes. There are two types of ribosomes within a eukaryotic cell. We have free ribosomes. You can kind of see them here where they're just floating in the cytoplasm. And then we also have ribosomes studded on the outside of one of the endoplasmic reticulum. The function of a ribosome is to assemble an amino acid chain. So it's one of the steps in making a protein. And it also has DNA. DNA is stored within the nucleus. The nucleus is here. The nucleus has a nuclear envelope, which is the outer layer of the nucleus. And then in that nucleus, there are pores, these little holes on the outside to allow whatever inside the nucleus to leave. So we need to have a couple of things leave the nucleus. One is ribosomes, because they are made within the nucleolus. The nucleolus is this small dark region here. This makes our ribosomes, and also the DNA is kept in the nucleus, and we have to make a copy of that DNA, and that copy is called RNA, and that will leave the nucleus through the pores. Chromatin is the form of DNA when the cell is doing its job. It will turn into chromosomes once it's going to start going through cell division. Leading out from the nucleus is the endoplasmic reticulum. Oftentimes you're going to find the rough endoplasmic reticulum right around surrounding that nucleus, and it will almost sometimes even attach to that nuclear envelope. And they are actually made of the same material. They are made of phospholipids, which is also very similar to that of the cell membrane. Endoplasmic reticulum, there are two types. We have the rough endoplasmic reticulum and the smooth. The rough endoplasmic reticulum has ribosomes studded on the outside, like I said earlier, and its job is to take the amino acid chain that is made on the ribosome and further process that protein. The smooth endoplasmic reticulum does not have ribosomes attached to the outside, and its job is to process lipids. Going back to the rough endoplasmic reticulum, once the proteins are processed within the rough endoplasmic reticulum, we need to get them packaged and sent out. So they are going to be sent from the rough endoplasmic reticulum to the Golgi apparatus. The Golgi apparatus is here. If you take a look at the Golgi, it kind of looks like a sandwich where the innards are squishing out. It often can be confused with the smooth endoplasmic reticulum, which looks more like a maze. The Golgi apparatus, its job is to package and sort proteins. It's going to take the protein from the RER and figure out where it needs to go, package it up, and then it will send it out of the cell. And then we have the mitochondria. The mitochondria is the powerhouse of the cell. This is where cellular respiration takes place. Cellular respiration takes glucose and breaks it down into ATP, which is our cellular energy molecule. The mitochondria is the power, the battery of the cell. Lysosomes are filled with enzymes, and they're there to break down old cell parts or to digest food. I like to think of this as the maid service of the cell. We also have, not pictured here, but very similar looking to a lysosome, is a vacuole, small vacuole. Vacuoles are used for storage of food or salts or water, and they're also used for moving particles throughout the cell. The other thing we have here is centrioles. Centrioles are part of the cytoskeleton system. They are made out of microtubules, and they are involved with the mitosis, or the replication of the cell. So when the cell divides, it provides tracks for the chromosomes to move on. They move on spindles that are partly provided by the centrioles. Centrioles are only found in animal cells. They are not found in plant cells. Now moving on to plants, you can see that a lot of the same organelles are in plant cells. We do have our nucleus. We have our endoplasmic reticulum, both smooth and rough. We have mitochondria. We have the Golgi. We have lysosomes. We have ribosomes. We have a vacuole, but in a plant cell, the vacuoles are a little bit different. The vacuoles of a plant cell are very large, and they're usually within the middle of the cell. This is filled with water, and it is there as a skeleton for the cell. It provides strength for that cell, kind of keeping that cell upright. And then one of the other differences is chloroplasts. Chloroplasts are very similar to that of mitochondria. Chloroplasts are double membraned. Mitochondria are also double membraned. They have their own DNA. Mitochondria also have their own DNA. And they can self-reproduce. So a chloroplast can create a new chloroplast, and a mitochondria can create a new mitochondria. This comes from a theory called the endosymbiotic theory. And this is where, way back in the day, when prokaryote cells were the only type of cell, there was a close relationship between two cells. One was a little bit larger than the other. And the larger cell engulfed, or basically brought in, the smaller cell within within itself, and instead of eating it, it used it to its advantage, either powering the cell. And this is how we think chloroplasts and mitochondria came to be, is one cell engulfing another and then using it for its advantage. The chloroplast does photosynthesis, and photosynthesis takes sun energy and stores it into glucose. Cells also have a cell wall, where animal cells do not. Plant cell walls are made of cellulose, which is that carbohydrate or polysaccharide. I want to take a closer look at the cytoskeleton, and this is found in both plants and animals. The cytoskeleton are a network that kind of creates a skeleton within the cell. It is made of microtubules and microfilaments. Looking at this picture here, we have microtubules and then microfilaments, and it creates a network within the cell. If you look at this picture over here, you can see the green is the cytoskeleton, the blue is the nucleus, and the red is the cell membrane. Also part of the cytoskeleton are flagella and cilia. Flagella, showed picture down here, are long tails used for forward motion. And the picture above is cilia. Cilia are small little hairs. Cilia can also be used for movement, but it also can be used, like in the human body, to move mucus within the airways. In your trachea, there are small little hairs on those cells that kind of beat back and forth. And what they do is they move the mucus so that way as it's catching debris when you're breathing, it can beat that mucus up and then you can swallow it so you can digest anything that you caught within that mucus before it gets into your lungs. And that is cells.